you that weren't with us at all yesterday, I want to welcome you to the Current Directions in Psychological Science Speaker Series, which is being presented by Pearson and APS. And now to start things off, I'm excited to introduce our first speaker of the day, Cynthia Bulik, who is the Distinguished Professor of Eating Disorders in the Department of Psychiatry in the School of Medicine at the University of North Carolina at Chapel Hill, where she is also Professor of Nutrition in the Gilling School of Global Public Health and the Director of the UNC Eating Disorders Program. A clinical psychologist by training, Dr. Bulik has been conducting research and treating individuals with eating disorders since 1982. Recently, she has applied information technology to the treatment of eating disorders and obesity to broaden the public health reach of our interventions. And her talk today will focus on the intricate dance of genes and environment in eating disorders.
So what we're going to do today is we're going to spend some time talking about the intricate dance of genes and environment in eating disorders. I'm going to take you through the three main eating disorders and then really help you try to wrap your head around how genes and environment interact to influence risk for these disorders. And I'll try to do it in a user-friendly way because sometimes the genetic research can become a little bit overwhelming. But basically what we're going to be talking about is anorexia nervosa, bulimia nervosa, and binge eating disorder. And each one of these has a different clinical presentation, a different clinical course, and affects a slightly different demographic. In the past, and when I started in this field years ago, anorexia and bulimia were considered primarily to be sociocultural disorders. There was a lot of time spent blaming families, saying that there were specific types of families that led to the development of eating disorders, which is patently untrue. There was a lot of time spent blaming society, which is still a very real issue, and when we get to the end of our talk, we'll talk about the role of environment in creating risk for eating disorders. And there was a lot of blame on the individual. People thought that somehow these were disorders of choice, and that people somehow chose to starve themselves or chose to binge and purge. Advance?
We're on anorexia nervosa.
I am not. Okay, so anorexia nervosa is marked primarily by low body weight. And what that means is that someone's weight is about 85% or lower below their ideal weight. Even when they're at this low weight, they have an intense fear of gaining weight. And one of the most perplexing features of anorexia is distorted body image. I'll show you what I mean by that in the next slide. But basically what we see is that individuals with anorexia often see themselves as large or part of their bodies as large, even when they are emaciated. They have extreme focus on their shape and weight. They tend to deny being ill, so they will say that everything's fine, even, for example, if they're 5'6" and weigh 65 pounds. Women who have anorexia often, but not always, develop amenorrhea, which means they stop having their menstrual periods for at least 3 months, and it affects about 1% of the female population, so about 1 in 100 people. The estimated female-to-male ratio is about 10 to 1. Next slide.
Distorted body image.
So what I think you're seeing now is a picture of a girl looking in the mirror, and she sees herself as having some flesh, so she's not emaciated. But if you click again, what you'll see, what we see when we look at this girl. And what you can see is that her image of herself is as much larger than she is in reality. We don't know if this is a perceptual disturbance or if this is a disturbance in top-down processing. It's one of the mysteries of anorexia. The next slide shows the age of onset of anorexia nervosa. And typically what we thought was the age of onset was around 15 to 19 years old. But we're seeing a complete shift in demographics in anorexia. So what we're seeing now is we're seeing more prepubertal cases, so young girls developing anorexia before they enter puberty, and we're also seeing more cases in midlife and late life women. Nicole, when I advance these, can you advance them? I apologize to the listeners, but it seems like I can advance things now.
What's happening is every time you say next slide, I'm hitting next for everyone that's viewing the webinar.
Okay, so are you seeing anorexia nervosa mortality now? That's where we want to be.
Yes.
Okay. So anorexia nervosa has the highest mortality rate of any psychiatric disorder. And I think this comes as a surprise to many people. The standardized mortality ratio is over 10, which means that an individual with anorexia is 10 times more likely to die from her illness than an age and sex-matched peer. And those deaths tend to be secondary to the illness and also suicide. So suicide is a severe risk in individuals with anorexia. Next slide. So when we talk about bulimia nervosa, we talk about the presence of recurrent binge eating. And a binge is eating an unusually large amount of food by social comparison in a short period of time, coupled with feeling out of control. And that's the key difference between overeating and binge eating, that notion of feeling out of control. The binges are coupled with compensatory behaviors, and that typically is either vomiting, excessive exercise, fasting, laxatives, or diuretics, which are water pills. Like anorexia nervosa, there's extreme focus on shape and weight, and it's a little more common than anorexia, afflicting about 2 to 3% of the population. But like anorexia, it is more common in females, although definitely is not exclusively found in females. Next slide. The next eating disorder is binge eating disorder. And binge eating disorder is marked by recurrent binge eating defined the same way as bulimia, but in the absence of those regular compensatory behaviors. So what that often means is that individuals with binge eating disorder are also suffering from overweight or obesity. And this can also happen in bulimia nervosa, but it's more commonly associated with binge eating disorder. They're distressed by their binge eating, and this is the most common eating disorder in the United States. So about 3.5% of people suffer from binge eating disorder. Next slide. A really important take-home point is that eating disorders don't discriminate. For a long time, eating disorders were believed to be exclusively in white upper-middle-class girls. This is completely untrue. These are only the people who were able to make it to some of the university-based treatment programs, and so there was a bias in terms of who people were seeing in their clinic. They afflict women, they afflict men, they afflict across racial and ethnic boundaries. And the current estimate is that about 9 million Americans are suffering from some form of an eating disorder. Next slide. So a couple important points. First off, eating disorders are not disorders of choice. They thought they were years ago. Part of when they talked about them as disorders of choice is it devalued the seriousness of the illnesses. It also undermined treatment and recovery because it didn't help families. Families would just say, if only she would eat more or if only she would eat less, not understanding the biological complexity of these illnesses. And when we first got into research on genetics of eating disorders, people thought that we were crazy because they thought if there's any psychological disorder for which genes weren't operative, it would be the eating disorders because the story that they were sociocultural illnesses was so plausible. Next slide. So let's talk about how genes and environment interact to influence eating disorders. Next slide. This slide that starts with genetic epidemiology outlines how we start to identify how genes play a role in psychological illnesses. So the first question that we ask is, do eating disorders run in families? And to do that, we do family studies. The thing about family studies is they can't help us unpack to what extent genes or environment influence whether something runs in families. Something could run in families because you watch your mother do it or you watch your father do it, or because he or she passed their genes on to you, or more likely some critical combination of both. So to unravel that question, we move on to twin and adoption studies. And then if twin and adoption studies suggest that genes play a role, then we can ask, where are the genes that influence this disorder and what do they do? And to do that, we move on to more molecular genetic designs that look at linkage, association, and genome-wide association studies. And I'll walk you through each three of these levels to help you understand what they mean. Next slide. So that first question, do eating disorders run in families? Next slide. So to do these studies, you basically look at a bunch of relatives of someone who has an eating disorder compared with a bunch of relatives of someone who doesn't have an eating disorder to find out if those relatives are at higher risk who have someone in the family with an eating disorder. And what we found is that anorexia and bulimia do run in families. In fact, relatives of someone with anorexia are 7 to 12 times at higher risk of having an eating disorder themselves than relatives of someone without an eating disorder. And that means that the relative risk of anorexia is as high as disorders like schizophrenia or bipolar disorder. But as I mentioned before, family studies can't tell you if this familial pattern is due to genes or environment. Next slide. So to tease that out, we do classical twin studies. And twin studies are really amazing because they're a natural biological experiment. And what they let us do is they let us take a large sample of twins, and sometimes we use samples as big as 40,000, 50,000. But they're made up of both monozygotic or identical twins who share 100% of their genes. They're basically clones of each other. And dizygotic twins who share on average 50% of their genes. So dizygotic twins are no more similar genetically than regular siblings, except unlike regular siblings, they also shared the womb and they shared their environment growing up. Next slide. So this is my cartoon to help you understand just overall how twin studies work. What we're looking for, in essence, is the frequency with which both members of a twin pair have the disorder. In this case, let's say anorexia nervosa, and that's called concordance. So if you hit next, you'll see some red lightning bolts striking. And if you hit next again, you're going to see some circles coming up. And what this cartoon is showing you is that monozygotic twins, where they're both dark blue, are more likely to be concordant for anorexia nervosa than dizygotic twins, where you see some sporadic lightning bolts where just one twin pair is affected. So whenever you see a pattern like this, where monozygotic twins are more likely to be concordant than dizygotic twins, that's your first suggestion that genes might be playing a role. And this is the kind of pattern that we've seen for eating disorders. Next slide. So this slide is titled Genes and Environment. And what we do is we go beyond that kind of a cartoon, and we break down the variance and liability to these disorders into genetic effects, which we call heritability. And just to be very clear from the beginning, if ever you read something that says the gene for anorexia was found or the gene for bulimia was found, that's completely wrong, because there will never be a single gene that causes any complex traits like eating disorders or depression or schizophrenia. A complex trait is defined as a trait that is influenced by many genes of small to moderate effect, as well as environmental factors. And there are two types of environmental factors. The first one is shared environmental effects. And those are environmental effects that act to make twins more similar, like the religion they grew up with, or a common parenting style, or their socioeconomic status. And then the second type of environmental effect is a unique environmental effect. Shakespeare would call this the slings and arrows of outrageous fortune. So this is something that one twin might experience, but the other might not. So one twin might have a soccer coach who's totally about strong bodies and women being really strong athletes. And the other one might have a ballet teacher who's all about get as thin as you can in order to be a beautiful live ballerina and go on a diet and use laxatives and do anything you can in order to lose weight. So those would be different, unique environments that make the twins more different. Next slide. What we found in these studies is not at all what anyone predicted we were going to find. And if you look at sort of the lavender bars, that tells you the A squared, or the heritability component for anorexia nervosa. And these four studies all show that the heritability of anorexia, that is the proportion of variance that's due to genetic factors is actually quite large, so somewhere between about 49 and 70%. That shared environment, back in the day when people said that this was a familial cause disorder, clearly accounts for only a very, very small proportion of the variance. And when environment does matter, it's actually those unique environmental factors. And unique environment also includes measurement error. So the next slide tells a similar story for bulimia nervosa. Just like anorexia nervosa, genes appear to play a considerable role in liability for bulimia nervosa. And finally, the next slide shows the same thing for binge eating disorder. And we have a couple of new studies that also confirm this, and that is that genetic factors also play a role in risk for binge eating disorder. So in the next slide, if you summarize across these twin study results, it tells us that genetic factors influence anorexia nervosa, bulimia nervosa, and binge eating disorder. It tells us that shared environment is not substantially contributory to risk for these disorders, but unique environmental factors are more important. These studies have now been replicated in several countries, but the one weakness is that they've all been done primarily on European ancestry populations. So we don't have the data about whether the same genetic and environmental mix seem to influence eating disorders in other racial and ethnic groups. Next slide. Okay, so if you remember our progression, the next step, once you determine that genes play a role, is to say, where are these genes and what do they do? So we started out with some candidate gene association studies, and that's where people just pick one gene and say, maybe this serotonin gene might be associated with anorexia. We've moved on from that to genome-wide association studies. I'm going to tell you what the difference is between the two of them, again in somewhat cartoon form, but to give you an idea of where we are in the genetics field. So in a single gene association study, you usually have a group of cases, several hundred cases and several hundred controls, and you're looking at one or a few genes. In a genome-wide association study, again you're comparing cases versus controls, but you're looking at around 1 million genetic markers scattered across the genome, so a much bigger search space. In a single gene association study, you have to make some guesses about what genes you think might be involved. In a GWAS, it's discovery science, so there's no guesswork. We're looking at all of them to see where the differences lie. In those single gene studies, we were looking at samples in the hundreds, and now in the GWAS studies, we're looking at samples in the thousands and tens of thousands. So what this picture is, the next slide, you'll see lots of dots. Those are 24,000 dots, and they represent all the genes in the human genome. Remarkably, the orange ones up top are the only ones we know anything about. The rest are still a mystery. We haven't figured them out yet. If you hit next slide, you should see one big red arrow coming up. When you do a single gene association study, you say, out of all those 24,000 genes, I'm going to study that one because I think it might have something to do with anorexia nervosa. And when you look at it this way, you realize that that's kind of absurd, given how many genes there are and how complex anorexia nervosa is. If you hit the next slide, you'll see this sort of like pink shading that covers nearly the whole genome. When you do a GWAS, this is the kind of coverage you get. And you're comparing your cases and controls, saying, of all of these genes that we're looking at, where do the differences lie? And then you correct for multiple comparisons and look for the most significant ones to show you the differences between your cases and controls. Next slide. Where we stand now is we're in the middle of doing the first large-scale genome-wide association study for anorexia nervosa. There have been some small ones done that haven't shown anything because they've been underpowered. But we've put together a consortium of 17 different countries. We have 4,000 samples, and the data are currently being analyzed in the United Kingdom. So with any luck, for relatively soon, we'll have some insight into possibly some ideas of what genes actually play a role in risk for anorexia nervosa. Next slide. What I don't want to do is make you think that this is all genetic. so we need to spend a little bit of time on the environment. But the environment can work both ways. The environment can exert both positive and negative influences on risk for eating disorders. Next slide. So here's some of the negative risks. Environment can increase risk if you are, for example, in sports with an appearance focus, or sports with a weight focus, or if you're heavily into dieting, unhealthy extreme dieting. Modeling has been implicated. People who are obsessed with looks and constantly focusing on their appearance. We also know that teasing is a risk factor for the development of both eating disorders and obesity. Next slide. Environment can also be a buffer, and it can help decrease the risk for eating disorders. So for example, family members can model healthy eating. Family members and friends can really help people learn how to separate their body esteem from their self-esteem. We can have positive role models for body respect, lots of family involvement, especially family breakfasts, supportive peers who value who you are, not how you look. But parents, of course, have to know that all of these environmental variables aren't necessarily controllable. Next slide. So let's take a look at how genes and environment interact. And again, this is a cartoon. So if we click next, in the middle, you're going to see an environment. And I put ballet up there as my high-risk environment. If you click again, you'll see two genotypes, little a, little b, and big A, big B, big B. Now, I call those little a, little b, my ballet vulnerable genotype, and the capital letters I call my ballet robust genotype. The point of this slide is that you bring your genes into an environment, and it's that critical combination of your genes and the environment that determine what come out the other end on the phenotype level, or what you look like. So if you click again, what you see is the person with the ballet robust phenotype managed to make it through ballet school and come out the other end a healthy ballerina. And if you click again, you see that that person who came in with the ballet vulnerable genotype succumbed to that environment, and it triggered her down an unhealthy path toward anorexia nervosa. So next slide. If you click again, you'll see a bunch of really colorful chromosomes. And these are the human chromosomes. Click one more time, and you're going to see a bunch of arrows going toward things like appetite regulation, harm avoidance, perfectionism, binge eating, obesity risk, activity level, obsessionality, ease of vomiting, and anxiety. These are all factors that we know are influenced by genes. Now, genes don't cause these things, but genes code for proteins that influence these things. So this first batch of arrows shows high-risk genetic factors. We're looking at 4 different parameters, starting with high-risk genetic. Click one more time, and you see genetic factors that buffer you. So genes influence self-esteem. Genes influence constitutional fitness. So genetic factors can also buffer you against eating disorders. Click again, and you see some lightning bolts. And these are the environmental risk factors. So for example, teasing and dieting. And if you click again, you see environmental buffering factors, like that really positive coach, family meals, or breastfeeding, which is good for just about everything. So it's this critical combination of genetic risk factors, genetic buffering factors, environment risk factors, and environment buffering factors that come together in this gene environment soup to influence your risk for developing an eating disorder. Next slide. And this is a simplified but worth thinking about comment, which is that genes load the gun and environment pulls the trigger. And that's really what we're thinking about in terms of the way that genes and environment interact to influence risk for eating disorders. Next slide. So here are the misperceptions, things that I don't want you to learn. First, genetic determinism. Just because you have high-risk genes doesn't mean you will get a disorder. As we talked about, there are buffering genes, there are risk environments, and there are buffering environments. And it's that critical combination that influences your risk. environment doesn't matter. In fact, if anything, environment matters more. The more genetic risk you are at, the more vulnerable you might be to these environmental insults. So it's really important that we still go out there and try to get more healthy models on the catwalks or find more healthy female role models who feel good about their bodies and can separate their body esteem from their self-esteem. And the third misperception is that biological disorders require pharmacological treatments. That is the etiology treatment fallacy, and it's just not true. There are plenty of cases in psychology and in medicine where psychosocial interventions actually do lead to change of fundamentally biological conditions. And that is the end of my presentation. Thank you very much for your attention.
We have our master teacher, Lynne Kemen, with us. Lynne, are you there?
Yes, I am. Hello, everybody.
Hey, all right. So our first question comes from an instructor, and I'd imagine it's a question that you might hear a lot. But the question is, how do you recommend instructors handle personal questions when they're asked in class? For example, I think my friend has an eating disorder. What do I do?
I usually answer that with, I would be happy to talk to you about it after class. I don't think that it's always appropriate for there to be sharing, and I also think that it's also important that if you are teaching a class in abnormal psychology, that the class understands that you are there to teach, that you're not there to conduct therapy sessions for them. And I think keeping that difference by announcing it right up at the very first class is really important. I think it's also important that you as an instructor have a master list of places where you can send people who do need counseling and get them to the right place for the correct referral. But I think it's not appropriate for the instructor to be doing that kind of a discussion in class.
Okay, thanks. And so those of you are aware, any questions for Dr. Bulik, you're welcome to ask and we'll answer them later. And any questions right now regarding teaching, we'll be answering live during the Q&A. So I'll take our next question for Dr. Kemen, which is, how do you teach your students to think critically about information that they might read about or hear about on the news or in the media?
Great question. This is something that you want to teach all semester, and I think part of it is putting it in a context for the students. They need to understand that we are very much a part. How we think and what we consider the truth and what we consider normal versus abnormal is very much a function of the time that we're in, of the society that we're in, of the particular culture that we're in, and that there's a vast continuum. I really like to talk in all of my classes about how perceptions or interpretations of conditions have been interpreted through the ages. When you look at some of the religious people during the Middle Ages, some of the nuns who were fasting, we're used to looking at it in a religious context, but you could start looking at it as an eating disorder. And that becomes a really interesting thing for students to sort of wrap their heads around. How weight and what the perceptions of what is considered beautiful are very much a part of the particular time that we're in. And I think we only have to go back as far as the 1950s, when we had people like Marilyn Monroe and Gina Lollobrigida who were size 12. And those were considered to be really beautiful people. Then in the 60s, we got into actresses like Twiggy, who was incredibly emaciated, and that became the norm of what was beautiful. And I think we can go back and look at photographs or paintings from different times and see what was considered beautiful, and we can see that we go all the way from probably size 18 to size -2 or -4, and it's just a matter of what particular time that we're in.
Okay, great, thanks. So helping them really to think critically about what they're reading and how it compares to today's society. So the next question is, again, related to the classroom and kind of that balance of, the research side of it and the clinical side of it. So the question is, my students often enjoy reading the case studies, but do you have suggestions for how to engage my students in the research aspect of the abnormal psychology material?
Well, I think Cynthia did a great job of sort of talking about some of the research and showing how we sometimes have ideas that this has a bigger effect than that, and showing the research that's going on, making people aware of the evolution of the thinking that's going on. I think it's really exciting. And I think her point about there is no single gene, I think very often we all sort of think, oh, well, there's a gene for schizophrenia, there's a gene for bulimia, and we don't put together what a complicated package it is. And making people aware that there's no one answer and no one gene, I think is part of what we're here to teach students.
Mm-hmm. Okay, and we have time for a couple more questions. The next one is, have you started introducing your students to possible changes to the upcoming DSM-5? And if yes, how have you done so?
Yes, again, this sort of goes back to the idea that this is an evolving kind of discussion. There was a time when homosexuality was considered to be a mental disease, and we laugh at that now, but it's been, certainly in my lifetime, that has changed. So I think making people aware that this is a work in progress and that what is considered normal versus abnormal is going to be changing a lot over time is something that we need to talk about and that we need to make people aware of. There aren't so many changes coming about in the DSM regarding eating disorders. The only one that I can think of is about PICO, which is where you are eating a substance that is not normally considered to be food. But I am assured that when DSM-6 comes out, the one after the one that we're expecting next year, that there will be further changes. And a lot of that will be a reflection of the research that's being done. And some of it was described for you just this afternoon.
Right, thanks. Yeah, so it's constantly evolving. Good point. So another question we have are just for specific suggestions for in-class activities that could help students better understand kind of that line of what's considered normal and what's considered abnormal.
Mm-hmm. Okay. First I want to say that the section on eating disorders is often the most painful and the most difficult for a lot of students to go through. I think there's a lot of shame and there's a lot of guilt about eating, and I think people need to sort of understand that it's not just about an eating disorder, that very often there are very complex emotional issues that are going on, that there may be biological or organic or medical issues that are going on. But in terms of students sort of understanding this, I think it's really good to have them talk about what our culture is showing us and what kinds of messages are out there about what the ideal body type is. I think you could also do things like have students keep a food journal and write down what they eat and when they eat it, and if it's not during a mealtime, or even if it is during a mealtime, what their particular emotional state was. Were they conscious about eating, or were they sitting in front of television and just eating and look down and go, oh, I've consumed my entire meal? Are they going over something that was upsetting that happened to them during the day? I wouldn't ask the student to turn it in. I think it's something that's private, but I think it can be really useful for sort of understanding that certain kinds of things trigger eating behavior. And it's not necessarily just that, oh, it's dinner time. And I think that's a good thing. I also like to make people, well, there was a point that Cynthia made about how it seems that a lot of eating disorders are starting younger and younger. I would bring in the fact that certainly young women, but also young males are starting to develop much earlier than they used to, and part of that is going to be because of the types of food that we're eating, about the hormones that are in the food. The weight problem that America is having right now, where we are considered to be mostly overweight and not in good shape, has a lot to do with lifestyle. It also has to do with the amount of corn syrup and sugar that is in all kinds of food that you might not expect it to be in. So making people aware of these factors, I think, is useful. And in my classes, at least, students really seem to enjoy talking about that.
Okay, thanks, Lynn. And we have a question related to the previous question that we were discussing on the new DSM. The question is, are you teaching now about the upcoming subcategory of purging disorder, or are you waiting until the new DSM comes out? And I don't have a background in this, so I don't know if you know more about this. I'm sure you do than I do. So,
I talk about things that are coming, or that we believe are coming, but I don't go into it in great depth because it's not out there yet. And I believe that some of these changes may still be in process, but I like to make people aware that different kinds of disorders and conditions are being broken down and thought about differently.
Really good point. Thanks. So last question, and you've touched on it in pretty much all of your answers, actually, because I know this is a sensitive topic, but the question is, how do you help really to destigmatize disorders as you present them in class?
The very first thing I do, and I do it in my syllabus, is try to create a space where people can speak about whatever is going on for them in a place where they will not be jumped on or laughed at or told that that's ridiculous. That idea of the continuum and what we think is normal is an ongoing lecture for me that I actually bring up. I think it would be unusual if there were a single class where we didn't go into that.
Mm-hmm. All right, well, that actually brings us to the end of our first session. So I want to thank you so much, Lynne and Cynthia, for just volunteering your time to share your research and your teaching strategies with all of us today. And for everyone on the line, we have a 15-minute break, so I hope you'll join us again when we resume at 1 o'clock Eastern, and our next topic is on behavioral neuroscience.